Hello Anatomists, Michele here, and today our video is all about the eye. So we're gonna do a series of videos, or one video with the models, and then I'll do a second video with diagrams. So to get started on this particular eye model, you can see we're looking at left eye. And we can get our orientation because we have the lacrimal gland on the top lateral side of the eye. And then we have what's called the lacrimal sac on the medial side. Okay, and so the lacrimal gland and the lacrimal sac are part of the lacrimal apparatus. That's going to help us when we look at the muscles that are on the eyeball itself. So right here is the lacrimal gland. So this is the top of the eye, and this muscle, remember from A&P1, is the superior rectus, straight muscle. And then remember that lacrimal sac is medial. So that would make this the medial rectus. Inferior rectus at the bottom, and then lateral rectus here. So we have 4 rectus muscles. Remember, rectus is talking about straight muscles. And then we have 2 oblique muscles. An oblique is like a curve, right? So when we're looking at the top of our model, we'll see on the top, we have a superior oblique. And then when we look at the bottom, we have an inferior oblique. Okay, now notice the inferior oblique is pointing towards that lacrimal sac, which remember is medial, right? Your tears drain down into your nose, right? When you cry, you blow your nose a lot. So lacrimal sac is medial. So if we look at this model here, we can see superior rectus, and we can see inferior rectus. And because the inferior oblique is curving around, that tells us that this, let me double check myself, I'm right, this is medial rectus. And then this would be lateral rectus. Now these external muscles of the eyeball are letting us look up and down and to the left and to the right. And when you're super annoyed, just rolling those eyes, baby. So we work those muscles hard when we're teenagers. Maybe you didn't, but I did. All right. Now those external muscles are attached to this white tissue of the eyeball, which is called the sclera. The sclera is part of the outer layer of the eyeball, which we call a tunic. And that outer layer, that outer tunic, is called the fibrous tunic. It includes not just the sclera, but also what we call the cornea. So the cornea, you can see on this model, is only shown on half. But the cornea is going to be this transparent part of the fibrous tunic that's going to be on the front of the eyeball. So it needs to be transparent so that the light can enter or penetrate. So with this model, I can take the top portion of the fibrous tunic off. And when I do that, I'm able to really see into what's called the vascular tunic. So when we're looking at our eyeball, you could think of it as like a 3-layered onion. So the outer layer, remember, fibrous tunic. The middle layer is the vascular tunic. And it's named vascular because this is where we see all of the blood vessels penetrating and servicing the tissue of the eyeball. Okay, so this bulk of this vascular tunic that we see here is called the choroid. When we're looking at the front, the iris, which is the colored part of the eye, it is a muscle that actually controls the diameter of the pupil. And so the iris and the choroid are part, this is easier if I do this, are part of that vascular tunic. Now take a look. The iris, I'm going to open it up, take out its guts there. The iris controls the opening, the diameter of the pupil. Pupil is just a hole, people. So this is like a structure that we're naming that's like not a structure because it's a hole. I always think that's super interesting. As I put it back together, we'll see that the pupil allows the light to penetrate to the lens. So also part of the vascular tunic is the structure called the lens, which we can see is this piece right here. The lens is going to be important, actually focusing the light onto our photoreceptor cells. And it's going to be like held in place by some ligaments, and those ligaments are controlled by a muscle. And so we'll see those structures better on our diagrams. This whole space, let me put it back together a minute. This whole space, once you're inside the cornea and you're in this space here in front of the iris, and then you're in this space all the way to the lens there, is going to be called the anterior cavity. And we'll do some more labeling on our diagrams to help with that. But that anterior cavity is full of a fluid, and that fluid is called aqueous humor. So the humor here is like an old-fashioned word for liquid. Aqueous, it means like watery. So we have an aqueous humor fluid, which is going to help support and nourish the tissue here that's located in the anterior chamber. When I pop off this part of the model, when I dissect, I showed you the lens here. The area behind the lens to the back of the eyeball is called the posterior chamber, and is full of a fluid, which is represented here. And this fluid is called vitreous humor. Now, the way I remember this is vitreous kind of sounds like the word viscous, and the word viscous means like thick. So like maple syrup, corn syrup, probably even better example, or honey, maybe even better example because it tastes super delicious. Honey is very viscous. So vitreous humor is going to be actually almost like holding its shape. It's so solid. So that's helping to hold the whole shape and support the shape of the eyeball itself. And that's filling up the posterior chamber. When we take a look then inside the model here, what we can see is in all of this tissue here, this is called the retina. The retina is part of our neural tunic. So this is actually where the photoreceptors are located. These are our sensory receptor cells, and they're synapsing onto our sensory neurons, which will make up our optic nerve, cranial nerve number 2. So the neural tunic is so named because this is where we start to see the sensation of vision being detected. Not processed really, but detected, okay? Now notice you have like this curly red line. That's going to be called the ora serrata, and that's going to be this division between our choroid and our our retina, right? Neural tunic is the big name for this layer. All right, when we look at the retina, we're going to see a very special place. You see it's kind of like highlighted in pink. This is called the Fovea Centralis. This is where you have your highest concentration of cone cells. Cone cells are our photoreceptors that detect color, and they give us like our best vision. So the reason we like things to be in color is because we can see it really clear when it's in color. So when we're looking directly at something, that wavelength of light should get focused directly on our highest center, our most accurate vision center, which is the Fovea Centralis. We already mentioned here the optic nerve, cranial nerve number 2, and if you notice, you have kind of like this circular area where that nerve is going to actually come out of the eyeball and into the brain. That area is going to be called the optic disc, and this is actually a place where we have a blind spot, so we're not actually able to detect if a wavelength of light lands in that particular area. Like, we don't see that. But we have 2 eyes, and so our visual spectrums overlap. So this is not something that we notice in our everyday life. And our brain really fills in a lot of gaps for us. So even a person maybe with one eye, their brain is helping to fill in some of those gaps, although clearly that individual is going to have compromised vision. Let me double check my list and see if we've covered all of our structures. All right, y'all. It looks like that's all we can detect on our models, so stay tuned for the next video with our diagrams. All right, take care of yourself and each other.